Hi everyone, how's it going? Got another lecture for you here. This is probably review for most of you. Certainly you biology majors, you probably started off taking cell biology, so we're not going to get into it as deeply as we do in cell biology, but you know, we're going to emphasize those things and review those things that are going to be necessary later on in the semester. And so you know, there's a lot of things to talk about. None of this is terribly difficult, so we split it up into two. So this is part one of cells. So this is a class about zoology. A lot of people are interested in zoology because you're interested in animals and organisms and big things. Why do I need to know this? Well, for the same reason that for our bio majors we have you take cell biology your first semester on here. Cells are the building blocks of all living things. If you want to understand biology, you have to understand cells. You've got to understand what's going on at the cellular level. I made this mistake when I was in your shoes when I was an undergrad. I was like, I'm an animal guy, I'm an organismal guy. Oh gosh, you know, and I had a bad attitude about it. And it took a long time for me to grow up and realize, oh, right. If I really want to have a deep understanding of biology, I have to understand cells. And so that's why we're talking about this topic today. And so this outline is going to be spread again across two separate lectures. We're going to break it up into two pieces. So let's get started. The first thing we're going to talk about is cell size. And we've got a little graphic here to kind of just put things into perspective. And so if you'll notice, this is a logarithmic graph. And so each division is an order of magnitude or a factor of 10, right? So we go from 10 meters down to 1 meter, down to 1/10 of a meter, so on and so forth. And this is sort of just to help you wrap your brain around what scale these different things are. And it's also to kind of briefly introduce the concept of microscopes and why we use microscopes and where do we which microscopes are appropriate for which things, and you know, in lab we're going to talk about microscopes a little bit. And so if you look, we're starting kind of at our scale, like a scale we can easily relate to, which is, you know, average human size, right? Between you know, 1 to 10 meters is sort of where you're going to find us. Then what they're showing their next, the next size scale is a nerve cell. And so this is a cell, a single cell, but it's a very special cell. When we talk about the nervous system, you'll understand why, but nerve cells are very long, but that's very atypical for cell sizes. And then you look at something like a chicken egg, would be the next scale down. And then you can compare chicken eggs to frog eggs, right? And so you've probably seen frog eggs. You've certainly seen chicken eggs. You can appreciate that they're both eggs, but of course the frog eggs are a lot smaller. But then human females also produce eggs, and they're an order of magnitude smaller. And so, you know, you can picture, maybe you've seen frog eggs at the side of a pond or something like that, but then, you know, an order of magnitude 1/10 that size would be a human egg. But all of these things are still big enough that you can see them with the unaided eye. But then when we go down even to smaller scales, that's where the eye can't resolve things that small. So the nerves and the receptors in your eye are not close enough together for you to be able to focus on something this small. But by using microscopes, we can focus on things smaller, and we can really start to understand things. And so here we've got human eggs and frog eggs that can be seen with the naked eye, but if you look at them under a microscope, you can see more detail. So then you get to the scale of 10 micrometers to 100 micrometers, and that's where we're working at when we're talking about eukaryotic cells. And so the cells we talk about today are going to be at that size. These are not something you can see with the naked eye, but you can easily see them and see their details with a regular microscope, a compound microscope. Then about 1/10 of that size are going to be things like bacteria and other prokaryotes, but also some of the organelles within your cells. So again, you can still see them with a microscope, but they're much, much smaller. And then smaller than that are the very small bacteria, and we get about 1/10 of the bacterial size. You're talking about viruses. And so these are going to be much smaller, and then as we go smaller, you're in the realm of macromolecules, and then small molecules, and down to the size of an atom, which is about 1/10 of a nanometer. And you can also see that we can resolve and look at some of these things with a light microscope, but also with an electron microscope. And electron microscopes are cool. The light microscope relies upon lenses to focus light better on your eye. The electron microscope, I'm sure, uses lenses, but also uses beams of electrons. And so their wavelengths are smaller than a wavelength of light, a wavelength of visible light. And so that allows you to resolve smaller and smaller things. And so we'll talk about microscopes in more detail later, but here's an example of a compound microscope image, and these would be typical cells, and you can see, so the things we're going to talk about today, you could all see with a compound microscope, not with the naked eye. But if you want to get smaller, again, you can look at electron microscopes. And so we're not going to get into electron microscopes, but I just think they're cool. We actually have an electron microscope out at the Hancock Biological Field Station, and we teach a class in electron microscopy. And so if you're interested in this sort of thing, it's really cool. But there's basically two types of electron microscopes: the transmission electron microscope and the scanning electron microscope. And they both allow you to see things, much, much smaller things. And so here's a picture from a transmission electron microscope, and you can see that the scale bar indicates we're in the order of about 2 microns, which is very, very small. But the real cool images come from a scanning electron microscope, because they're 3D, and they look very cool. So if I back up, this is a cilia, the little finger-like projections on the inside of your intestine. And here's the same cilia, so it's the same structure, but you can see the difference. You can see the 3D structure. So these are really cool. And this is what we have out at Hancock. We've got a scanning electron microscope. Now what's really cool about the scanning electron microscopes is, well, obviously the stuff you can see. And in fact, there are competitions where people submit their best scanning electron pictures. And so for no other reason than I think they're cool, I've shown you some of these. And so these are pollen grains, and it's just, I just think it's cool because, again, this is an incredibly small things, but you can see the incredible detail in them. And so it just makes you think about, all the cool stuff you can see in the world, but if you shrink yourself down, there's even just as much cool stuff. Here's the antenna from a cockroach, right? Look at the detail you can see. That's so cool. This is a close-up of a nematode. This is an organism we're going to talk about in lab, a roundworm. And you can see the details of the mouth that it uses to attach itself to the intestines and anchor itself. This is a scale from a lungfish. Something we talk about in my ichthyology class is that you've got these little microscopic bumps. These actually make the fish more hydrodynamic. You trap a boundary layer of water next to the fish, and so you think something rough would actually make you move through the water, inhibit, you would think being rough would inhibit your movement through the water, but actually being rough at the microscopic scale actually enhances your hydrodynamic efficiency. So this is pretty cool. Can you, sounds like a bunch of geese getting ready to land on the pond behind me. Nope. They took a look at the pond and they went right over. They'll probably circle back though. My neighbor has 3 pairs of geese that all had babies this year. So they come down and they visit our pond every once in a while. All right, sorry for that. So that's a scale from a lungfish. This is the wing of a mosquito. So again, just think about that. You know what a mosquito is. You've seen a mosquito, but you've never probably thought about what the detail is like on its wing. This is so cool. This is what a human hair looks like, so this might help kind of wrap your brain around what scale we're looking at. So if you zoomed in on one of your hairs, this is what it would look like. This is a bunch of E. coli bacteria growing on a surface. Just kind of cool looking. And this is the most metal thing ever. This is the head of a flea. And look how awesome that is. I mean, that is crazy, right? And again, you've seen a flea, but you never had any idea they looked this cool. So anyway, I just wanted to show you that because we're talking about cells, which need to be looked at with microscopes, and this is a type of a microscope, and there you go. So I've made the point that cells are small, and so now let's ask the question, well, why? Why are cells small? So the membrane doesn't stretch too much? Because they're easy to replicate when they're small? To maintain a high surface area to volume ratio? Or to minimize the oxygen needed for respiration? Well, the answer is C. It's all about surface area to volume ratio. And this is a concept that comes up again and again in biology. So let's investigate it in a little bit more detail. So if I ever talk about SAV, or write that acronym down, I'm talking about surface area to volume ratio. So how much surface area or covering you have relative to the inside, relative to the total volume. And surface area is a two-dimensional measurement. Volume is a three-dimensional measurement. Well, the surface area to volume ratio is critical to the function of a cell. As a cell gets bigger, the surface area increases by a factor of 2, but the volume increases by a factor of 3. This is true for all three-dimensional structures. As things get bigger, their volume gets bigger faster than their surface area. So consequently, as things get bigger, their surface area to volume ratio goes down. Small cells have a high surface area to volume ratio compared, and big cells have a low surface area to volume ratio. And it's the surface area to volume ratio that determines how efficiently the cell can function. So let's look at an example, a simple example. Cells are spheres, but we can illustrate the concept by looking at cubes. And so here we've got some different situations, and we're going to compare them to one another. So the first column is just a single small cube, and it's got, a unit size of 1, say 1 centimeter on a side. It doesn't matter what the units are, but let's say 1 centimeter. And so if you calculated the total surface area of that cube, each side is 1 square centimeter, and there are 6 of them, so the total surface area would be 6 square centimeters, right? The volume, the length times the width times the height, would be 1 cubic centimeter, right? And so the surface area to volume ratio is 6 to 1, or 6. Now, imagining that's a cell, we look at the evolution of organisms over time. They started off as small, single-celled organisms, but eventually became bigger. Now, there's a couple of ways that an organism can get bigger, right? You start off as a single cell, and you all started off as a single cell, right? Think about that. All of us at one point, your dad's sperm fused with your mom's egg, and you were a single cell. And from that, you eventually grew into a very large organism, but we were all a single cell at one point. And so you could get bigger in one of two ways. You could be a single cell, and that cell just gets bigger and bigger and bigger and bigger. Or you could make many copies of the cell, and you could get bigger by making more copies of the small cell, right? There's two ways. So which way is more efficient? Let's talk about if you just have the one cell, and that cell gets bigger and bigger and bigger, and that's represented by the center column. And so in this example, we've got a bigger cell, or a bigger cube, and this thing is 5 centimeters on a side. So the surface area of one side would be 5 times 5. It'd be 25 square centimeters. And you've got 6 of those, and so the total surface area is 150 square centimeters. The volume of this cell is 5 times 5 times 5. It's 125 cubic centimeters. So you have a lot more surface area in this bigger cell. But you also have a lot more volume, right? So you've got 25 times as much surface area, but you've got 125 times as much volume. Consequently, the surface area to volume ratio for this big cell is 1.2. Big things have very low surface area to volume ratios. But let's take the second strategy. We want to get bigger. So instead of being one cell and the cell just gets bigger, let's just have many cells and the cells stay small and there's just a lot more of them. What happens to the surface area to volume ratio? So now we're getting to the same size. If you look in the third column, that structure is the exact same size as the one in the center column, but it's made up of lots of small boxes. Each of those boxes is 1 centimeter on a side. So now let's look at the total surface area. And so you've got lots and lots of boxes. You've got each one of those boxes, the surface area is 6 square centimeters. But there's so many boxes, the total surface area is 750 square centimeters, right? So you got to take the surface area of each one of those little boxes. The total volume is the same as the center column. It's 125 cubic centimeters. So again, we're occupying the same volume, right? But we're breaking it down into smaller bits. We've got 125 little cells, and each of those cells has 6 square centimeters of surface area. So the surface area to volume ratio of this third column is 6. It's a much higher surface area to volume ratio. So the point is, we have made something, the center column and the last column, these two organisms are the same size, but if you do it the last way by making small cells, you get a much higher surface area to volume ratio. Okay, well, why then is that important? That a high surface area to volume ratio is important to cells, but why? Because things have to get into and out of cells, right? Every cell has to take in oxygen and has to get rid of carbon dioxide. Every cell has to take in nutrients from your food and then has to get rid of the waste, right? That's how cells function, is they take in things and they get rid of things. Well, how do you take in things? You get them, they have to cross the surface area. They have to cross the outer edge of that cell. That's surface area. So if you have more surface area, that's more places where things can get into the cell. But when they get into the cell, they have to get to every part of that cell. So stuff has to get all the way to the center of that cell, and stuff produced in the center of that cell has to get out, right? So you need a lot of area to get things in, but once they get in, you don't want them to have to travel very far. And so you need a small volume. So you need a high surface area relative to the volume. And so that's kind of what I'm saying here. With a low surface area to volume ratio, it's tough to get things all the way from outside the cell all the way to the middle of the cell. There's not as much surface area that you can cross, and when you do cross, you got a long way to travel. Whereas if you've got a high surface area to volume ratio, once things cross that membrane, they don't have to travel very far. And so that makes cells more efficient when they're small. Okay, well, this concept of surface area to volume ratio is going to come up in a lot of places, but it's very important to cells. All right. So let's start talking about the parts of the cell, and that surface area is defined by the cell membrane. And so let's talk about the plasma membrane or the cell membrane. This, of course, is what creates the outer edge of the cell. It acts as a selective barrier. And so it doesn't make any sense to have an outer barrier that just lets everything in and lets everything out, right? The cell couldn't function as a cell. You have to make the inside of that cell different from the environment. That's what life is all about. Living things are different from their environment. If you're the same as your environment, then you're just a piece of the environment. And so it has to let some stuff in, but not everything. Now, this is a zoology class, and so we're just concerned about animal cells. But we, of course, we have to talk about plants too, right? All life begins by capturing sunlight. So plants are also very important. Plants have a cell wall on the outside of this cell membrane. So all living things have a cell membrane, but plants also have the cell wall on the outside. Now, we've talked a little bit already about the cell membrane when we talked about lipids. And of course, the cell membrane is made of a phospholipid bilayer, right? So you've got two layers of phospholipids, and they're back to back to each other. They've got that hydrophilic head, which is made of a phosphate, and that sticks to the outside of the cell and to the inside of the cell. And then those hydrophobic tails are tucked to the inner part of the cell membrane, which you can see here. And that's because the inside of the cell, the cytoplasm, has water. The environment around the cell has water. So those hydrophilic heads interact with that water, but those hydrophobic tails are trying to tuck into the center of the membrane to get away from the water. But it's not just, phospholipid bilayer, that's the basis of it. That's the foundation. But there's a lot more in the cell membrane. And of course, if you had cell biology, you talked about all this, and there's a lot more going on. But basically, you can see there's lots of things studded and sticking into this membrane. And some of those things just stick on the outer surface, and some of those things just stick on the inner surface, and some of those things go all the way through the membrane. And it's not a rigid membrane. All these molecules can kind of flow around, and so that's why it's called the fluid mosaic model, right? It's not rigid, things can move, that's the fluid part. The mosaic is when you have pieces of different things that all come together. One of the things that gives the cell membrane its fluidity are those cholesterol molecules. And so if you look at this diagram, you can see where it's indicating cholesterol. And again, we talked about that when we talked about lipids. The cholesterol is a type of lipid, it's a steroid, and too much cholesterol in your diet can lead to cardiovascular disease, but you've got to have cholesterol because you need it for your cell membranes. And you can see lots of other proteins and things that are studded in this cell membrane. So, we're going to be seeing this a lot throughout the semester. Now, some molecules can go right through the membrane. It doesn't keep everything out. And there are certain things that can get right into the cell very quickly. But most things have to, if they want to get into the cell, they have to go through some kind of a hole that's put into the cell membrane. And those holes are usually made by proteins, some sort of protein channel. And so this is an idea that's going to be real important to explain a lot of things in this class. Proteins that allow certain stuff in and exclude other things. Now you also see that they've labeled some of these proteins are glycoproteins. And so a glycoprotein is just what it sounds like. It's a carbohydrate plus a protein. It's a combination of two types of molecules. A lot of times these are important on the cell membrane because they identify self versus non-self. And so this is a way that your immune system can identify which cells belong to you, and then can also identify which cells belong to a foreign invader, so they know what to attack. So these membranes are not just for the outer edge of the cell. Don't worry about me. I live out in the country, so gunshots are not a problem most of the time. And it's just my neighbor practicing, no big deal. So the membranes are not just for the outer edge of the cell. A eukaryotic cell also has internal membranes. And those internal membranes define many of the organelles that we're going to learn about. And so here's a figure from your book. You're very familiar with this. So study these. You've probably seen a million of these. This is a three-dimensional, typical view of the cell. Here's just another one. And so we're going to talk about a lot of these organelles in part 2. And so there's lots of organelles. Again, you probably talked about these before. We're not going to go into tremendous detail. These are the ones that you need to know and understand their basic function. And so we'll go over these in part 2. So take a look at those diagrams and prepare yourself for part 2, and I'll be back in just a little bit with part 2 of our tour of the cell. So see you then.